3. Plant Kingdom. In the previous chapter, we looked at the broad classification of living organisms under the system proposed by Whitaker in 1969, wherein he suggested the five kingdom classification, which is Monera, Protista, Fungi, Animalia, and Plantae. In this chapter, we will deal in detail with the further classification within the kingdom Plantae, popularly known as the plant kingdom. We must stress here that our understanding of the plant kingdom has changed over time. Fungi and members of a Monera and Protista having cell walls have now been excluded from plants through earlier classification placed them in the same kingdom. So, the cyanobacteria that are also referred to as a blue-green algae are not algae anymore. In this chapter, we will describe algae, bryophytes, pteridophytes, gymnosperms and angiosperms under Plantae. Let us also look at the classification within the angiosperm to understand some of the concerns that influenced the classification system. The earliest system of a classification used only gross superficial morphological characters such as habit, color, number and shape of leaves, etc. They were based mainly on a vegetative characters or on the androecium structure, means system given by Linnaeus. Such systems were artificial. They separated the closely related species since they were based on a few characteristics. Also, the artificial system gave equal weightage to the vegetative and sexual characteristics. This is not acceptable since we know that often the vegetative characters are more easily affected by the environment. As against this, natural classification system developed, which were based on a natural affinities among the organisms and consider not only the external feature but also the internal features like ultrastructure, anatomy, embryology and phytochemistry. Such a classification of the flowering plants was given by George Bentham and Joseph Dalton Hooker. At present, phylogenetic classification system based on the evolutionary relationships between the various organisms are acceptable. This assumes that the organisms belonging to the same taxa have a common ancestor. We now use information from many other sources to to help resolve difficulties in classification. This becomes more important when there is no supporting fossil evidence. Numerical taxonomy, which is now easily carried out using computers, is based on all observable characteristics. Numbers and codes are assigned to all the characters and the data are then processed. In this way, each character is given equal importance and at the same time hundreds of characters can be considered. Cytotaxonomy, that is based on cytological information like chromosome number, structure, behavior, and chemotaxonomy that uses the chemical constitutions of the plants to resolve confusions, are also used by the taxonomists these days. 3.1 Algae. Algae are the chromophil Algae are the chlorophyll bearing simple thalloid autotrophic are largely accepted 3.1 Algae. Algae are chlorophyll bearing, simple, thalloid, autotrophic and largely aquatic, means both freshwater and marine organisms. They occur in a variety of other habitats, moist zones, soils and woods. Some of them also occur in the association with the fungi as lichen and animals, example on a sloth bear. The form and size of algae is highly variable, ranging from the colonial forms like volvox and the filamentous forms like Eulothrix and Spirogyra, figure 3.1. A few of the marine forms such as kelps form a massive plant bodies. The algae reproduce by vegetative, asexual and sexual methods. Vegetative reproduction is by fragmentation. Each fragment develops into a thallus. Asexual reproduction is by the production of different types of spores, the most common being the zoospores. They are flagellated, means motile, and on germination gives rise to a new plants. Sexual reproduction takes place through the fusion of two gametes. These gametes can be flagellated and similar in size as in Eulothrix, or on non-flagellated, means non-motile but similar in size as in Spirogyra. Such reproduction is called isogamous. Fusion of two gametes dissimilar in size, as in the species of Eudorina, is termed as anisogamous. Fusion between one large non-motile means static female gamete and a smaller motile male gamete is termed as oogamous. Example, volvox and fucus. Algae are useful to man in a variety of ways. At least half of the total carbon dioxide fixation on the earth is carried out by algae through photosynthesis. Being photosynthetic, they increase the level of dissolved oxygen in their immediate environment. They are the paramount importance as primary producers of energy rich compounds which form the basis of food cycles of all aquatic animals. Many species of Porifera, Laminaria and Sargassum are among the 70 species of marine algae used as a food. Certain marine brown and red algae produce large amounts of hydrocolloids, means water holding substances. Example, algine means brown algae and carrageen in red algae, which are used commercially. Agar, one of the commercial products obtained from the Gelidium and Gracilaria, are used to grow microbes and in preparations of ice creams and jellies. Chlorella, a unicellular alga rich in proteins, is used as a food supplement even by space travelers. The algae are divided into three main classes: Chlorophyceae, Phaeophyceae and Rhodophyceae. 3.1.1 Chlorophyceae. The members of Chlorophyceae are commonly called green algae. The plant body may be unicellular, colonial or filamentous. They are usually grass green due to the dominance of pigments chlorophyll A and B. The pigments are localized in a definite chloroplast. The chloroplasts may be discoid, plate-like, reticulate, cup-shaped, spiral or ribbon-shaped in the different species. Most of the members have one or more storage bodies called pyrenoids located in the chloroplast. Pyrenoids contain protein besides starch. Some algae may store food in the form of oil droplets. Green algae usually have a rigid cell wall made of an inner layer of cellulose and an outer layer of the pectose. Vegetative reproduction usually takes place by the fragmentation or by the formation of different types of spores. Asexual reproduction is by the flagellated zoospores produced in a zoosporangia. The sexual reproduction shows considerable variation in the type and formation of sex cells, and it may be isogamous, anisogamous or oogamous. Some commonly found green algae are Chlamydomonas, Volvox, Ulothrix, Spirogyra and Cara. Figure 3.1a. 3.1.2 Phaeophyceae. The members of Phaeophyceae or brown algae are found primarily in the marine habitats. They show great variation in size and form. They range from simple branched filamentous forms like Ectocarpus to profusely branched forms as represented by kelps, which may reach a height of 100 meters. They possess chlorophyll A, C, carotenoids and xanthophylls. They vary in color from olive green to various shades of brown depending upon the amount of xanthophyll pigment fugoxanthin present in them. Food is stored as a complex carbohydrates which may be in the form of laminarin or mannitol. The vegetative cells have a cellulosic cell wall usually covered on the outside by the gelatinous coating of algae. The protoplast contains, in addition to plastids, a centrally located vacuole and a nucleus. The plant body is usually attached to the substratum by holdfast and has a stalk, the stipe and leaf like photosynthetic organ, the frond. Vegetative reproduction takes place by fragmentation. Asexual reproduction in most of the brown algae is by biflagellate zoospores that are pear-shaped and have two unequal laterally attached flagella. Sexual reproduction may be isogamous, anisogamous or oogamous. Union of gametes may take place in water or within the oogonium, means oogamous species. The gametes are pyriform, means pear-shaped and bear two laterally attached flagella. The common forms are Ectocarpus, Dictyota, Laminaria, Sargassum and Fucus. Figure 3.1b. 3.1.3 Rhodophyceae. The members of Rhodophyceae are commonly called red algae because of the predominance of the red pigment R phycoerythrin in their body. Majority of the red algae are marine with greater concentrations found in warmer areas. They occur in both well-lighted and regions close to the surface of water, and also at the great depths in the ocean where relatively a little light penetrates. The red thalli of the most of the red algae are multicellular. Some of them have a complex body organization. The food is stored as a Floridian starch, which is very similar to the amylopectin and glycogen in structure. The red algae usually reproduce vegetatively by fragmentation. They reproduce asexually by non-motile spores and sexually by the non-motile gametes. Sexual reproduction is oogamous and accompanied by the complex post-fertile developments. The common members are Polysiphonia, Porphyra, Gracilaria, and Gelidium. Table 3.1, Division of algae and their main characters. Please take a look at the table. 3.2, Bryophytes. Bryophytes include the various mosses and liverworts that are found commonly growing in a moist shaded areas in the hills. Figure 3.2. Bryophytes are also called amphibians of the plant kingdom because these plants can live in soil but are dependent on water for sexual reproduction. They usually occur in a damp, humid and shaded localities. They play an important role in a plant succession on bare rocks or soil. The plant body of bryophytes is more differentiated than the algae. It is thallus-like and the prostrate or erect, attached to the substratum by unicellular or multicellular rhizoids. They lack true roots, stem or leaves, but they may possess root-like, leaf-like and stem-like structures. The main part of a plant body of the bryophyte is haploid. It produces gametes, hence it is called gametophyte. The sex organs in bryophytes are multicellular. The male sex organ is called antheridium. They produce biflagellate anthrazoids. The female sex organ called archegonium is a flask-shaped and produces a single egg. The anthrazoids are released into water where they come in contact with the archegonium. An anthrazoid fuses with the egg to produce the zygote. Zygote do not undergo reduction division immediately. They produce a multicellular body called sporophyte. The sporophyte is not free living but attached to the photosynthetic gametophyte and derives nourishment from it. Some cells of the sporophyte undergo reduction division, meiosis, to produce haploid spores. These spores germinate to produce gametophyte. Bryophytes in general are a little economic importance, but some mosses provide a food for herbaceous mammals, birds and other animals. Species of sphagnum, a moss, provide peat that have long been used as spores. These spores germinate to produce gametophyte. Bryophyte in general are of a little economic importance, but some mosses provide food for herbaceous mammals and animals. Packing species of sphagnum, a moss, provide peat that have long been used as fuel and as a packing material for transshipment of a leaves material because of their capacity to hold water. Mosses, along with the lichens, are the first organisms to colonize rocks and hence are a great ecological importance. They decompose rocks, making the substrate suitable for the growth of higher plants. Since mosses form dense mats on the soil, they reduce the impact of falling rain and prevent the soil erosion. The bryophytes are divided into liverworts and mosses. 3.2.1 Liverworts. The liverworts grow usually in moist, shady habitats such as banks of streams, marshy ground, damp soil, bark of trees, and deep in woods. The plant body of a liverwort is a thalloid, example Merchantia. The thallus is a dorsiventral and closely appraised to the substrate. The leafy members have a tiny leaf-like appendages in two rows on the stem-like structures. Asexual reproduction in liverwort takes place by the fragmentation of thalli or by the formation of specialized structures called gemae, singular gemma. Gemae are green, Multicellular asexual buds which develop in small receptacles called gamma cups located on the thalli. The gamma become detach from the parent body and germinate to form a new individuals. During sexual reproduction, male and female sex organs are produced either on the same or a different thalli. The sporophyte is differentiated into food seta and capsule. After meiosis, spores are produced within the capsule. These spores germinate to form free living gametophytes. 3.2.2 Meiosis. The predominant stage of a life cycle of a moss is in the gametophyte, which is consist two stages. The first stage is a protonema stage which develops directly from the spore. It is creeping, green, branched and frequently filamentous stage. The second stage is a leafy stage which develops from the secondary protonema as a lateral bud. They consist of a upright, slender axis bearing spirally arranged leaves. They are attached to the soil through multicellular and branched rhizoids. These stages bear the sex organs. Vegetative reproduction in mosses is by the fragmentation and budding in a secondary protonema. In a sexual reproduction, the sex organs Antheridia and Archegonia are produced at the apex of a leafy shoots. After fertilization, the zygotes develops into sporophyte consisting of a food, seta and capsule. The sporophyte in mosses is more elaborate than the liverworts. The capsule contains spores. Spores are formed after meiosis. The mosses have an elaborate mechanism of a spore dispersal. Common example of mosses are Funaria, Polytrichum and Sphagnum, figure 3.2. 3.3 Pteridophytes. The pteridophytes include horsetails and ferns. Pteridophytes are used for medicinal purposes and as a soil binders. They are also frequently grown on ornamentals. Evolutionarily, they are the first terrestrial plants to possess vascular tissues, xylem and phloem. You shall study more about these tissues in chapter 6. The pteridophytes are found in a cool, damp, shady places, though some may flourish well in a sandy soil conditions. You may recall that in bryophytes, the dominant phase in the life cycle is a gametophyte plant body. However, in pteridophyte, the main plant body is a sporophyte which is differentiated into true root, stem and leaves. Figure 3.3. This organ possesses well differentiated vascular tissues. The leaves in the pteridophyta are small, means microphyles, as in Selaginella, or large macrophyles, as in ferns. The sporophytes bear sporangia that are subtended by leaf-like appendages called sporophylls. In some cases, sporophylls may form distinct compact structures called strobili or cones in Selaginella equisetum. The sporangia produce spores by meiosis in a spore mother cells. The spores germinate to give rise to inconspicuous, small but a multicellular, free-living, mostly photosynthetic thalloid gametophytes called prothallus. These gametophytes require cool, damp, shady places to grow. Because of this specific restricted requirement and need for a water for fertilization, the spread of living pteridophytes is limited and restricted to the narrow geographical regions. The gametophyte bear male and female sex organs called Antheridia and Archegonia respectively. Water is required for the transfer of anthrazoids. The male gametes released from the antheridia to the mouth of archegonium. Fusion of a male gamete with the egg present in archegonium result in the formation of zygote. Zygote thereafter produces a multicellular, well-differentiated sporophyte, which is a dominant phase of pteridophyte. In majority of the pteridophytes, all the spores are similar in kinds. Such plants are called homosporous. Genera like Selaginella and Salvinia, which produce two kinds of spores, macro means large and micro means small spores, are known as heterosporous. The megaspores and microspores germinate and give rise to female and male gametophyte respectively. The female gametophytes in these plants are retained on the parent sporophyte for a variable periods. The development of a zygotes into young embryos takes place within the female gametophyte. This event is a precursor to the seed habit, considered as an important step in evolution. The pteridophytes are further classified into four classes: Psilopsida, Psilocytum, Lycopsida contain Selaginella, Lycopodium, Spinopsida contain equisetum, and the Pteropsida contain Dryopteris, Pteris, and Adiantum. 3.4 Gymnosperms. The gymnosperms means gymnos means necked and sperma means seed are plants in which the ovules are not enclosed by any ovary wall and remains exposed both before and after fertilization. The seed that develops post fertilization are not covered, that is are necked. Gymnosperms include medium sized trees on tall trees and shrubs. Figure 3.4. One of the gymnosperms, the giant redwood tree Sequoia, is one of the tallest tree species. The roots are generally tap roots. Roots in some genera have fungal association in the form of mycorrhiza in Pinus, while in some other means cycas, small specialized roots called coralloid roots are associated with the N2-fixing cyanobacteria. These stems are unbranched, cycas, or branched in Pinus and Cedrus. The leaves may be simple or compound. The cycas, the pinnate leaves persist for a few years. The leaves in a gymnosperms are well adapted to withstand extreme of temperature, humidity, and wind. In conifers, the needle-like leaves reduce the surface area. Their thick cuticle and sunken stomata also help us to reduce water loss. The gymnosperms are heterosporous. They produce haploid microspores and megaspores. The two kinds of spores are produced within the sporangia that are borne on the sporophylls, which are arranged spirally along an axis to form a lax or a compact strobili or cones. The strobili bearing microsporophylls and microsporangia are called the microsporangiate or a male strobili. The microspores develop into male gametophyte generation, which is highly reduced and is confined only a limited number of cells. This is reduced gametophyte is called the pollen grain. The development of pollen grain takes place within the microsporangia. The cones bearing the megasporophylls with ovules or a megasporangia are called macrosporangiate or a female strobili. The male or a female cones or strobili may be born on a same tree means Pinus. However, in cycas, male cones and megasporophylls are born on a different trees. The megaspore mother cell is differentiated from one of the cells of a nucellus. The nucellus is protected by the envelopes and the composite structure is called an ovule. The ovules are born on a megasporophylls which may be clustered to form as the female cones. The megaspore mother cell divides meiotically to form 4 megaspores. One of the megaspores enclosed within the megasporangium develops into multicellular female gametophyte and bears 2 or more archegonia or female sex organs. The multicellular female gametophyte is also retained within megasporangium. Unlike bryophytes and pteridophytes, in gymnosperms, the male and female gametophytes do not have an independent free living existence. They remain within the sporangia written on the sporophytes. The pollen grain is released from the microsporangium. They are carried in air currents and come in contact with the opening of the ovules born on a megasporophylls. The pollen tube carrying the male gamete grows towards the archegonia in the ovules and discharge their contents near the mouth of the archegonia. Following fertilization, zygote develops into an embryo and ovules into seeds. These seeds are not covered. 3.5 Angiosperms. Unlike the gymnosperms, where the ovules are necked, in the angiosperms or flowering plants, the pollen grains and ovules are developed in the specialized structure called flowers. In angiosperms, the seeds are enclosed in fruits. The angiosperms are exceptionally large group of plants occurring in a wide range of habitats. They range in a size from the smallest wolfia to tall trees of eucalyptus over 100 meters. They provide us a food, fodder, fuel, medicines and several other commercially important products. They are divided into two classes, the dicotyledonous and monocotyledonous. Figure 3.5. The ultimate summary. Plant kingdom includes the large Plant kingdom includes algae, bryophyte, pteridophytes, gymnosperm, and angiosperm. Algae are chlorophyll-bearing, simple thalloid, autotrophic, and largely aquatic organisms. Depending on the type of pigment possessed and the type of stored food, algae are classified into three classes, namely Chlorophyceae, Phaedophyceae, and Rhodophyceae. Algae usually reproduce vegetatively by fragmentation, asexually by the formation of different types of spores, and sexually by the formation of gametes which may show isogamy, anisogamy, or oogamy. Bryophytes are the plants which can live in a soil but are dependent on water for sexual reproduction. Their plant body is more differentiated than that of algae. It is thallus-like and prostrate or erect and attached to the substratum by rhizoids. They possesses root-like, leaf-like, and stem-like structures. The bryophytes are divided into liverworts and mosses. The plant body of liverworts is a thalloid and dorsiventral, whereas mosses have a upright, slender axis bearing spirally arranged leaves. The main plant body of a bryophyte is a gamete-producing and called a gametophyte. It bears male sex organs called antheridia and female sex organs called archegonia. The male and female gametes fuse to form zygote, which produces a multicellular body called a sporophyte. It produces haploid spores. The spores germinate to form gametophytes. In pteridophytes, the main plant is a sporophyte which is differentiated into true root, stem, and leaves. These organs possesses well differentiated vascular tissues. The sporophytes bear sporangia which produce spores. The spores germinate to form gametophytes which require cool, damp places to grow. The gametophytes bear male and female sex organs called antheridia and archegonia respectively. Water is required for the transfer of male gamete to archegonium where zygote is formed after fertilization. The zygote produces a sporophyte. The gymnosperms are the plants in which ovules are not enclosed by an ovary wall. After a fertilization, the seeds remain exposed, and therefore these plants are called naked seeded plants. The gymnosperms produce microspores and megaspores, which are produced in a microsporangia and megasporangia borne on a sporophylls. The sporophylls, microsporophylls, and megasporophylls are arranged spirally on axis to form male and female cones respectively. The pollen grain germinates and the pollen tube releases the male gamete into the ovule where it fuses with the egg cell in archegonia. Following fertilization, the zygote develops into embryo and the ovule into seeds. The angiosperms are divided into two classes, the dicotyledonous and monocotyledonous. Thank you.